Hello everyone, I'm Yanping. I would like to introduce my project. In fact, I just focus on one neglected species in the family Emergomyces. Based on our previous study, it has proved that Emmonsia is a close neighbor of the novel genus Emergomyces. Here. And traditionally, this genus, Emmonsia, included two classic species, Emmonsia crescens and Pava, with the latter as a type species. But based on our previous study, has proved these two species lie in different clades. Moreover, our morphology data has proved Emmonsia Pava could produce big eel-like seal and the same as its neighbor Blastomyces. So, this data give us more support to separate these two species and rename Emmonsia Pava to Blastomyces. But with the name disappear, a bigger problem is coming. That the name of Emmonsia crescens should not be valued according to the rule of nomenclature. So, how can I determine the taxonomy of this species? So, let's go back to this phylogeny tree, and Emergomyces is close sister. So, what's the relationship between the two populations? We can see, in fact, neither pathogenic phase and the host is totally different. So, how can we determine the relationship between the two? The rivalry between the two genera, or it's possible to involve the two into one genus. And here, sorry, what? Oh, we can. Here we can. The starting point here. Emmonsia crescens comprise the two species, two subclades. And one just from Eurasian, another from North American, just always associated with geographic related. So that's the aim of the study. We just would like to investigate whether there is sufficient evidence to support the intraspecific variation in this species, and how important to the variation to this generic taxonomy. Okay, our study just carried out for the following aspect. First, let me introduce phylogeny analysis. First, we can see all strains. We just only 23 strains, all clustered into two subclades, sub-main population, especially RBB2 demonstrate strong supportive for the two population. Here we can see the blue part represent North American and the gray part represent global. And our AFLP genotyping analysis almost consistent with our phylogeny analysis as well. And following, we preferred haplotype and tree analysis. Both of these studies totally consistent with our phylogeny analysis, and the two studies demonstrate two main populations with no more than 30 mutations. Here we can see mutations just very few. The most distinct from our phenotypic studies. We can see from this graphic, the average adiaspore of every species in this part, in red part. This part represents global, and the diameter almost be over 30 microns. But we can see this blue part. This means this present North American group. Most of strains just demonstrate very, very relative smaller adiaspore with a diameter almost no more than 30 microns. And here, this graph just very clear to demonstrate the difference between these two populations and demonstrate the procedural adiaspore development of these two populations. Its top one is from global and the bottom one is from North American. We can see here, even cultured enough time, the adiaspore in this North American always produce smaller adiaspore and couldn't continue to expand. And here, that's the striking here. We can see big is the seal like seal in this population. That's really to our traditional views. And then following, how about the relationship between the host animal and the grouping? In fact, since our samples are very few, but we just get the review, prefer the review, the literature review, we can see, in fact, the geographic related grouping actually derived from the geographic distributed of the host animal. And more important, our study demonstrates the relationship between the frequency of adiaspore and the body temperature of the host animal. So it seemed the more frequency and the lower body temperature, that's amazing. For example, here, this column represents highest frequency of adiaspore, and the corresponding animal, that's wildwood. And this small rodent always keep low body temperature and always keep almost 33 degrees. And here, we can see this just a very few post-founding of adiaspore, and the corresponding animal, just this one, very cute one. And that's a European rodent. And these mammals always keep high body temperature, always almost reach 40 degrees. So taken together, first, based on our combined gene analysis, we can see Emmonsia crescens and the single strain Emmonsia sorting and the genus Emergomyces all involved one monophilic clade with strong bold trip spot here. And secondly, based on our morphological analysis, we can see Emmonsia crescens not only produce big adiaspore, but but also could produce adiaspore like seal, and big is like seal. That's amazing. And to the genus, very, very famous genus now, because they always cause human infection. And normally, pathogenic phase is smallest, but the members of this genus, for example, the type species of the genus, could produce big yeast as well. That means there is exact overlap between the two populations. It seemed the tendency from the adiaspore to small yeast. Then at last, from the ecological side aspect, we can see the two, both of them share the same habitat and the same biological behavior. Both of them can inhale the colider from the soil and cause infection or colonization. So above this point, we propose cluster Emmonsia crescens and the single strain Emmonsia sorting to one genus Emergomyces. And we believe with more samples added, the taxonomy of possession Emmonsia crescens should be more clear and precise. But until now, we believe that's the best option. So thanks a lot.
Thank you very much for a fantastic presentation. Congratulations. Do we have some questions? I would have one, but I will wait on others Do we have some questions on online streaming? No? In audience? Do we have some questions? No, we don't have a question. Okay, so I would have one. The Emmonsia is quite pathogenic. Are you feeling never a kind of fear by going to the lab and working with this pathogenic fungus? What is about your feelings when you're coming into the touch with this strong pathogen? It's in BSL-3 category, if I'm not right. If I'm right.
How to say, in fact, Emmonsia crescens not 3, not the top one, and so not very dangerous, just belong to 2. So it's okay to me. But to another, because I focus on this work, including another type of fungi, that's really a little bit dangerous. But because I finished this experiment in CBS, and here is this is standard lab, so it's okay. And I think most important, just I love this work. Yeah.
Thank you very much. So.